Hello and welcome to Chapter 16, Inherited Change. Okay, so oblong-winged katydids are normally green and well camouflaged, but sometimes a bright pink katydid occurred. So the pink color is caused by an allele of the gene that determines body color. It has always been thought that the allele causing the pink color was recessive, but breeding experiments in 2013 showed surprisingly that this is a dominant allele. A katydid that has one allele for pink color and one for green color is pink. So as we go through the chapter, think about how breeding experiments with katydids could have determined whether the pink color is caused by a dominant or recessive allele. So homologous chromosomes. So remember that homologous chromosomes are pairs of chromosomes that contain the same type of genes but maybe different alleles. So they have similar size, similar shape. You get one from mom and one from dad. Okay, and this is a picture of a karyogram. It's where your chromosomes are quite literally pictured and lined up. So you can see that each of the pairs that you're looking at are homologous pairs of chromosomes. You can see 22 autosomal chromosomes or non-sex determining chromosomes, and then you have the sex chromosomes at the bottom that are placed separately. So you can talk about your genes from time to time, like, oh, I have this gene or that gene, but how do we actually see them? A gene is a functional unit of DNA, and your DNA is organized into chromosomes. Chromosome banding is a little like tie-dyeing your chromosomes. A band is defined as that part of a chromosome which is clearly distinguishable from its adjacent segments by appearing darker or brighter with one or more banding techniques. There are 22 matching pairs of chromosomes. These are called homologous chromosomes. The word homologous means similar in structure and composition. Each pair is given a number. In the original zygote, one of each pair came from the mother and one from the father. So there is also a non-matching pair labeled X and Y. There are therefore two sets of 23 chromosomes, one set of 23 from the mother and one set of 23 from the father. The non-matching X and Y chromosomes are the sex chromosomes, and they determine the sex of the individual. All the other chromosomes are called autosomes. It is conventional to position the two sex chromosomes to one side in a karyogram so that the sex of the organism can be recognized quickly. So in humans, females have two X chromosomes and males have one X and one Y. The Y chromosome has portions missing and is therefore smaller than the X chromosome, and they're missing evolutionarily. Like the Y chromosome, which can only come from a male, has gotten smaller over time. Each chromosome has a characteristic set of genes which code for different features. Scientists are gradually identifying which genes are located on which chromosomes and what their precise functions are. So for example, we now know that the gene for the genetic disorder cystic fibrosis is located on chromosome 7. The gene for a particular characteristic is always found at the same position or locus on a chromosome. Each chromosome typically has several hundred to several thousand gene loci, many more than shown. The total number of different genes in humans is thought to be about 20,000 to 25,000. Each member of a homologous pair possesses genes controlling the same characteristics. So a gene for a given characteristic may exist in different forms, and that's called alleles. Okay, and alleles are expressed differently. So for example, the gene for eye color has two forms, one coding for blue eyes and one for brown eyes. So you'll have a gene for eye color, but two alleles, an allele for blue and an allele for brown. So an individual could possess both alleles, one on the maternal chromosome and one from the paternal. So mom might pass down the allele for blue and dad might pass on the allele for brown. Okay, and this is an image from your book. This just shows some of the genes that we know about. It shows on the left there the normal expression, and then on the right some of the disease or genetic disorders that can be caused by faulty expressions of those genes. And again, these are just some of the ones that we found. Okay, so these are looking specifically at the X chromosome. So this is showing the effects of normal and faulty expression. Homologous chromosomes are a pair of chromosomes in a diploid cell that have the same structure as each other, with the same genes, but again, not necessarily the same alleles, at the same loci, and that pair together to form a bivalent during the first division of meiosis. A gene is a length of DNA that codes for a particular protein or polypeptide, and an allele is a particular variety of that gene. So a locus is the position at which a particular gene is found on a particular chromosome. The same gene is always found at the same locus. Okay, so here you see homologous chromosomes carry the same genes at the same loci. And here you're looking at just 7 genes labeled A through G. And in reality, obviously, there's hundreds or thousands on each chromosome. But this shows a diploid cell with 2 complete sets of chromosomes, and you can see the banding there, each banding representing a different gene on homologous chromosomes. So when animals other than humans are looked at, we still find that cells usually contain 2 sets of chromosomes, and such cells are described as diploid. Again, so a cell that has 2 sets of chromosomes is described as diploid, and this is represented as 2n, where n equals the number of chromosomes. And not all cells are diploid. Gametes have only 1 cell of chromosomes. A cell which contains only 1 set is described as haploid, and this is represented as n. In humans, normal body cells are diploid, so autosomal cells all over your body are diploid. They have 2 sets of chromosomes with exactly 46 chromosomes, and gametes are haploid, meaning they have only 23 chromosomes or 1 set. So when a diploid zygote, which is 1 cell, grows into an adult with millions of cells, the new cells must be genetically identical with the same number of chromosomes as the cells that divide to produce them. The type of nuclear division that achieves this is called mitosis. So for the life cycle to contain sexual reproduction, there must be a point where fertilization takes place, and the number of chromosomes is halved. So this results in gametes containing only 1 set of chromosomes rather than 2 sets. If there were no point in the life cycle when the number of chromosomes halved, then the number would double every generation. And so the type of nuclear division that halves the chromosome number is called meiosis. And gametes are always haploid as a result of meiosis. Meiosis is sometimes described as reduction division because the number of chromosomes is reduced in the process. Meiosis brings genetic variation into the gametes. That way it's not just like a process of cloning, and therefore zygotes are produced. Genetic variation also arises as a result of mutation, which we'll talk about later, and that can occur at any stage in a life cycle. Such variation is the raw material on which natural selection has to work, and it has produced a huge range of species that live on Earth. So here is a model that is showing the process of meiosis. So if you look at this at the top, this is a model showing you how meiosis doesn't work. And basically what it's showing you is that after fertilization, you have a zygote that has 2 sets of chromosomes, 1 from mom and 1 from dad. And as that zygote grows into an adult, it does so through mitosis, right? That's what this little star shows. It shows that the zygote, which is 1 cell, has to grow to an adult cell with millions of cells. Therefore, mitosis has to occur so that cloning of each cell can happen. Okay, and then at that point, then reproduction would occur again. But if there wasn't reduction division, in other words, if this adult didn't produce gametes that had only 1 set of chromosomes, or if they did not produce haploid gametes, then each generation would double. So then this generation would have 96 chromosomes, and it would continue to double. But that isn't what happens. Okay, this is a model of what actually happens, where you start with haploid gametes. A one-cell zygote is created. That zygote multiplies through mitosis to equal an adult with millions of cells. Okay. Then through the process of meiosis, that adult produces haploid gametes, which after fertilization give you a one-cell zygote with the right set of chromosomes, that is the amount that we actually have, that we expect to see, which is 46 chromosomes in the zygote and in the adult. Okay, so meiosis brings, unlike mitosis, meiosis involves two divisions called meiosis 1 and meiosis 2. Meiosis 1 is a reduction division resulting in two daughter nuclei with half the number of chromosomes of the parent nucleus. And you will be required to know the steps of meiosis, all the way from meiosis 1 to meiosis 2. So we're looking at early prophase, middle prophase 1, late prophase 1, metaphase 1, anaphase 1, telophase 1, and then in meiosis 2, the chromosomes behave as in mitosis, so that each of the two haploid daughter nuclei divide again. And meiosis therefore results in a total of 4 haploid nuclei, okay, that you'll see through anaphase and telophase. Okay, so you are responsible for knowing these phases of meiosis. Okay, so what is independent assortment? The principle of independent assortment describes how different genes independently separate from one another when reproductive cells develop. So the law of independent assortment speaks of alleles of a gene separating independently from alleles of another gene. Hence, the inheritance pattern of one trait will not affect the inheritance pattern of another. For instance, the gene for eye color is inherited independently from the gene for hair color. So as recombination is happening during meiosis, and pieces of the chromosomes from mom and dad are assorting together, hair color and eye color will sort independently. So one gamete might contain the alleles for brown hair and blue eyes, and another gamete from the same process might contain the alleles for brown hair and brown eyes. A process occurring during meiosis wherein two chromosomes pair up and exchange segments of their genetic material is called crossing over, a direct cause of genetic diversity among cells produced in meiosis. So during prophase 1, homologous chromosomes pair up and become attached to one another. And at this time, crossing over moves sections of DNA between homologous chromosomes and allows for independent assortment. And this is what allows different siblings to look different from their parents, right? Because in each of those gametes, there's a different combination of those homologous chromosomes. Otherwise, siblings would look identical to each other, and they would all be the exact same mix of their mom and dad, but that's not what we see. Okay, so here's a picture of that. Homologous chromosomes are aligned, crossing over occurs, and then recombination occurs. So you might have your mother's eyes and your father's nose, but your brother has your mother's nose and your father's eyes. Sometimes a brother and sister can be full siblings with the same two parents, yet they can look pretty differently. And nobody can necessarily guess that siblings, what they're going to get, right? What combination of traits they're going to get. So why does this happen? Why aren't all siblings identical? That's because physical traits are manifestations of our genes. And while studying inheritance, the famous geneticist Gregor Mendel discovered that genes are inherited independently of each other. Hence the independent assortment. Okay, so the concept defines the law of independent assortment. And the formation of male gametes is known as spermatogenesis, and the formation of female gametes is oogenesis. So sperm production takes place inside tubules in the testes. Here, diploid cells divide, so that's important to remember. We're talking about the production of gametes in the male. Diploid cells divide by mitosis to produce numerous diploid spermatogonia, which grow to form diploid primary spermatocytes. The first division of meiosis takes place, forming 2 haploid secondary spermatocytes. The second division of meiosis produces haploid spermatids, which will mature. Okay, so here again is that picture of the creation of gametes. You have a diploid cell, okay, and you go through reduction to division to equal 4 haploid gametes. Okay, so this takes place inside the ovaries, where diploid cells divide by mitosis to produce many oogonia. These begin to divide by meiosis, but stop when they reach prophase 1. At this stage, they're called primary oocytes, and they are, of course, still diploid. All of this happens before a child is born, and at birth, a female will have 400,000 primary oocytes in her ovaries. So when she reaches puberty, some of the primary oocytes still get a little further with their division by meiosis. They proceed from prophase 1 to the end of the first meiotic division, forming 2 haploid cells. However, the division is uneven. One cell gets most of the cytoplasm and becomes a secondary oocyte, while the other is a little more than a nucleus and is called a polar body. The polar body can be thought of as simply a way of getting rid of half of the chromosomes and has no further role to play in reproduction. So it's not like the production of sperm in that way. Each month, one secondary oocyte is released into the oviduct from one of the ovaries. If it is fertilized, it continues its division by meiosis and can now be called an ovum. The chromosomes of the spermatozoon and the ovum join together to form a single diploid nucleus, and the cell that is made by this process is called a zygote. The zygote can now divide repeatedly by mitosis to form an embryo and then a fetus. Okay, so looking at gametogenesis in flowering plants, male gametes are produced in the anthers and female gametes in the ovules. Inside the anthers, diploid pollen mother cells divide by meiosis to form 4 haploid cells. The nuclei of each of these haploid cells then divide by mitosis, but the cell itself does not divide, resulting in cells that each contain 2 haploid nuclei. These cells mature into pollen grains, each surrounded by a protective wall made up of a tough exene and thinner intine. One of the haploid nuclei is called the tube nucleus, and the other is the generative nucleus. And these are the male gametes. Inside each ovule, a large diploid spore mother cell develops. This cell divides by meiosis to produce 4 haploid cells. All but one of these degenerates, and the one surviving haploid cell develops into an embryo sac. That will grow larger, and its haploid nucleus divides by mitosis 3 times, forming 8 haploid nuclei. And one of these becomes the female gamete. Okay, this is a picture from your book. Here you can see the ovary and the ovule. Okay, so moving on to genetics. So a gene is a length of DNA, like we talked about earlier, that codes for the production of a polypeptide molecule. The code is held in sequence of nucleotide bases in the DNA. A triplet of 3 bases codes for 1 amino acid in the polypeptide that will be constructed on the ribosomes in the cell. One chromosome contains enough DNA to code for many, many polypeptides. Okay, this is a review for you. The gene that codes for the production of beta globin of the hemoglobin molecule is on chromosome 11. Each cell contains 2 copies of the gene, one from mom and one from dad. There are several forms of varieties of this gene. One variety contains the base sequence GGACTTCTC and codes for normal beta globin. Another variety contains the base sequence GGACATCTC and codes for a different sequence of amino acid that forms a variant of beta globin known as sickle cell. Okay, different varieties of the same gene are called alleles. Most genes, including beta globin, have several different alleles. So for the moment, we will consider only the above 2 alleles of this gene. And for simplicity, the different alleles of a gene can be represented by symbols. In this case, they can be represented as follows. So there's the beta. This is the allele for the normal beta globin polypeptide here. And then here's HBS, which is the allele for the sickle cell B globin polypeptide. HB stands for the locus of the hemoglobin gene, and the superscripts A and S stand for particular alleles of the gene. Okay, in a cell, which is diploid, there are 2 copies of beta globin. The 2 copies might be HB to the A, or HB to the S, or HBA and HBS. So the alleles that an organism has form its genotype, and in this case we are considering just 2 different alleles. There are 3 possible genotypes, right? Because if you get this gene from mom and this gene from dad, or you could get this gene from mom and this gene from dad, or you could get a combination of the 2, one from mom, one from dad, right? So there's 3 different possible genotypes, even though we're only considering 2 different alleles. So a genotype in which the 2 alleles of a gene are the same is said to be homozygous for that particular gene, right? So here we're seeing that this individual is homozygous for HB to the A. A genotype in which the 2 alleles of a gene are different, for example, this one, is said to be heterozygous for that gene. And the organism can also be described as homozygous or heterozygous for that characteristic. So genotype affects phenotype. Okay, so a person with the genotype HB to the A and HB to the A has 2 copies of the gene in each cell coding for the production of the normal beta globin polypeptide. So all of this person's hemoglobin will be normal, and this person is said to be homozygous for the normal type beta globin polypeptide. Okay, a person with the genotype HB to the S, HB to the S, has 2 copies of the gene in each cell coding for the production of sickle cell. Okay, this person is also homozygous, but this person is homozygous for the sickle cell variant. All of this person's hemoglobin will be sickle cell hemoglobin, which is insufficient at transporting oxygen. This person will have sickle cell anemia. A person with the genotype HB to the A and HB to the S has 1 allele of the beta globin gene coding for the production of the normal beta globin and 1 for the sickle cell. So half of the person's hemoglobin will be normal and half will have the sickle cell. Such people have sickle cell trait and are sometimes referred to as carriers. They will probably be completely unaware that they have sickle cell trait because they have enough normal hemoglobin to carry enough oxygen. So for the most part, probably won't have problems. They may appear to be perfectly healthy. And difficulties arise only occasionally. For example, if they go to a place with high altitudes or do really strenuous exercise, then concentrations in the blood might become low. The observable characteristics of an individual are called a person's phenotype, and we will normally use the word phenotype to describe just the 1 or 2 particular characteristics that we are interested in. So for an example, if you look at this moth over here, the genotype might be big A, big A, or little a, but the phenotype is the color in this case. So dark, or light, or I'm sorry, the shade, not the color. Okay, so if we're looking at genotype for sickle cell, it would be here. So either homozygous normal, heterozygous, or homozygous sickle cell. But the phenotype is going to be what actually plays out from those genes in the physical form. So the person that's homozygous for the normal beta globin is going to have a normal phenotype. The phenotype for heterozygous is going to be normal, but with a sickle cell trait. And the phenotype for homozygous sickle is going to be sickle cell anemia. In sexual reproduction, haploid gametes are made following meiosis from diploid body cells. Each gamete contains one of each pair of chromosomes, therefore each gamete contains only one copy of each gene. Meiosis of a heterozygous cell produces gametes of two different genotypes. So, if you are a heterozygous individual, meaning that you have sickle cell trait from mom and normal beta globin from dad, this is the original diploid cell that's going to go through the process of meiosis to create gametes, right? But because you have an you have a mix of the two types for mom and dad, your gametes will also have a mix. So some gametes will have the sickle cell trait and some will have the normal. As there are equal numbers of each type of sperm and each type of egg, the chances of each of these four possibilities are also equal. So each time a child is conceived, there is a 1 in 4 chance that it will have the genotype homozygous normal, a 1 in 4 chance that it will be homozygous sickle cell, and a 2 in 4 chance that it will be heterozygous. Okay, and this is if we're looking at a carrier father and a carrier mother, right? Two heterozygous individuals. They're going to have a 1 in 4 chance of having normal, 1 in 4 of carrier, another 1 in 4 of carrier, and then a 1 in 4 of full-blown sickle cell anemia. So another way of describing these chances is to say that the probability of a child being homozygous for sickle cell, the probability of being homozygous for normal, and the probability of being heterozygous is 0.25 to 0.25 to 0.5. So it's important to realize that these are only probabilities, though. It would not be surprising if this couple had two children, both of whom had the homozygous for sickle cell and so suffered from sickle cell anemia. Right? It's just like the probability of flipping a heads on a coin that has heads on one side and tails on the other is 50%, but you can flip several times in a row and get heads each time, right? So we're only talking in terms of probabilities. So a genetic diagram is the standard way of showing the genotypes of offspring that might be expected from two parents. Okay, I'm going to give you an example concerning snapdragon flowers of different colors. Okay, so one of the genes for flower color has two alleles, namely CR, okay, which gives red flowers, and CW, which gives white flowers. What color flowers would be expected if the offspring from a red and a pink snapdragon? So you would expect about half of the offspring to have red flowers and about half to have pink flowers. Okay. So you can see here are the possible genotypes. Here are the phenotypes. Here is the diagram for a genetic diagram for a cross between a red and a pink. Okay, so the phenotypes are red and pink. The genotypes are homozygous for red and heterozygous for pink. Okay. All of the possible gametes that this parent can contribute is red, right? But the possible gametes that this parent can contribute are red or white in equal proportions. So offspring genotypes and phenotypes. you do a little Punnett square here. So you have one parent here. Okay, this is the parent. These are the gametes from the pink parent. You've got red and white, possible. And then here are the gametes from the red parent. You've got red, possible. When you put those together, you have homozygous red and homozygous pink. I'm sorry, heterozygous pink. So half of the offspring, and again in terms of probability, will be red and the others will be pink. So in the case of sickle cell anemia, both of the alleles in the heterozygous organism have an effect on the phenotype. Okay, a person with some normal hemoglobin and some sickle cell hemoglobin, a snapdragon with the genotype of CRCW has some red color and some white color so that the flowers appear pink. Alleles that behave like this are said to be codominant. Okay, frequently, however, only one allele has an effect in a heterozygous organism, and this allele is said to be the dominant allele, whereas the one that has no effect is recessive. And an example would be stem color in tomatoes. So there are two alleles for stem color. One will produce a green stem and the other a purple stem. In a tomato plant that has one allele for purple stems and one for green, in other words, they're heterozygous, the stems are exactly the same shade of purple as in a plant that has two alleles for those purple. Okay, the allele for purple stems is dominant. So even if you have an allele for green and an allele for purple, because purple is dominant, that is the color that's going to show in the phenotype. When alleles of a gene behave like this, their symbols are written as capital letter for the dominant and a small letter for the recessive, and you're often free to choose the symbols that you use. So in this case, the symbols could be A for the purple gene, capital A for the purple gene, and little a for the green allele. The possible genotypes and phenotypes for stem color are here. Okay, so you have homozygous for stem color is purple, and it's important to say homozygous dominant for stem color, right? Because you can have two homozygous pairings, one here and one here. Okay, this is homozygous recessive, this is homozygous dominant, and this is heterozygous. Okay, a dominant allele is one whose effect on the phenotype is identical to its effect in a homozygote. A recessive allele is one that is only expressed when there's no dominant allele present. And in codominant alleles, both have an effect on the phenotype. So the pink and the white giving pink. The red and the white giving pink. So the F1 generation is the offspring resulting from a cross between an organism with a homozygous dominant genotype and one with a homozygous recessive genotype. The F2 generation is the offspring resulting from a cross between the two heterozygous organisms. Okay, so F1 generation here is the offspring resulting from a cross between a homozygous dominant and a homozygous recessive. Okay, so here's your F1, and then your F2 is the offspring resulting from a cross between two heterozygous parents. Okay, so here's another example. You have homozygous for tall, homozygous for short, giving all heterozygous offspring in that F1 generation. And then a cross between those heterozygous offspring will yield, very predictably, the probability of having 3 tall, okay, and 1 short. Let's look at that a little bit more. Where alleles show dominance, it is not possible to tell the genotype of an organism. Right, because you could be heterozygous or homozygous in showing the dominant characteristic. So a purple-stemmed tomato plant might have the genotype capital A, right, because purple is dominant, or it could have the genotype capital A, little a. So to find out its genotype, you can cross it with a green-stemmed tomato plant to see what their offspring look like. So from the colors of the offspring, you can tell the genotype of the parent. If any green offspring are produced, then the purple plant had to have been heterozygous. This cross is called a test cross. A test cross always involves crossing an organism showing the dominant phenotype with one that is homozygous recessive. Okay, so looking at this purple stem tomato plant's genotype, okay, So the potential, so we're going to look at the two potentials. One potential is that that purple plant has a dominant dominant homozygous for dominant. So then by breeding it with a homozygous recessive plant, you would get all purple offspring, right? Because the female here can only give a capital A, right? So the only gamete passed down on this side is the capital A, and the capital A will always cause a purple phenotype. But here, if that purple plant, that original purple plant, the parent, is heterozygous, and it's crossed with a homozygous recessive green plant, the purple could give a capital A, the dominant A, or a recessive A, meaning that it could have a heterozygous offspring that's purple, as well as a homozygous offspring that's green. So if there is a green offspring, we know that this purple parent gave a small a. So far we've considered just two alleles, or varieties of any one gene. Most genes have more than two alleles, though. So an example of this situation known as multiple alleles is the gene for human blood groups. So the four blood groups, A, B, and O, are all determined by a single gene. Three alleles of this gene exist. Okay, you can see it here. IA, IB, and IO, and of these, IA and IB are codominant, whereas IO is recessive to both. Color variations in rabbits caused by multiple alleles of a single gene. Okay, so you can see here's multiple alleles for the color in rabbits. Okay, you have dominant, and then these recessive. Okay. So in rabbits, a series of multiple alleles controls coat color. In humans, sex is determined by one of the 23 pairs of chromosomes. These chromosomes are called sex chromosomes. The other 22 are called autosomes. The sex chromosomes differ from the autosomes in that two sex chromosomes in a cell are not always alike. They do not always have the same genes in the same position, and they are not always homologous. This is because there are two types of sex chromosomes, known as X and Y. So Y chromosome is much shorter than the X and carries fewer genes. A person with two X chromosomes is female, whereas a person with one X and one Y is male. This brings us to sex linkage. The X chromosome contains many different genes. One of them is a gene that codes for the production of a protein needed for blood clotting called factor VIII. There are two alleles of this gene, the dominant one producing normal factor VIII and the recessive one resulting in a lack of factor VIII. The recessive allele causes the disease hemophilia, in which the blood fails to clot properly. The fact that the gene for hemophilia is on the X chromosome and not on the autosome affects the way that it's inherited, right? Because females who have two X chromosomes have two copies of that gene. And males only have one X chromosome, so they only have that one copy. Therefore, the possible genotypes for men and women are different for that allele. The factor VIII gene is said to be sex-linked. Okay, so a sex-linked gene is one that is found on a part of the X chromosome not matched by the Y, and therefore not found on the Y. Genotypes including sex-linked genes are always represented by symbols that show that they are on the X chromosome. So the genotype of a woman who has the allele H on one of her X chromosomes and the allele H on the other is written as X to the H and X to the little h. Okay, so then you can see here is the cross between the normal man and a carrier woman. So if you look at these genotypes, it's X to the capital H and Y. Here we have X to the capital H and X to the lowercase h. Okay, so both parents can give different gametes. And then so this is the probability of offspring from the pairing between those two individuals. Dihybrid crosses look at the inheritance of two genes at once. Tomato plants, stem color A, purple, stem a green, a green stem. Okay, leaf shape, D-cut leaves, and then also little d, potato leaves. So in the heterozygote, due to independent assortment and meiosis, there are 4 possible gamete combinations. So let me say that again. We're looking at a dihybrid cross, which is the inheritance of 2 genes at one time. So what are going to be the offspring of parents that have combinations of these traits? Either the color, which is capital A, purple dominant, lowercase a, recessive green, and leaf shape. So the cut leaves or the potato leaves. Okay, so there are 4 possible combinations. So you're going to have dominant for color and leaf shape, heterozygous for color and shape, heterozygous for color and shape, recessive for color and shape. The plant with green stem and potato leaves must have the genotype as homozygous recessive. Each of its gametes will contain 1 A allele and 1 D allele, both lowercase. All of the gametes will have the genotype lower a, lower d. At fertilization, any of the 4 types of gametes from the heterozygous parent may fuse with the gametes from the homozygous parent. So from this test cross, therefore, we would expect approximately equal numbers of the 4 possible phenotypes, right? A 1-to-1 to 1-to-1 ratio is typical of a dihybrid cross between a heterozygous organism and a homozygous recessive organism, where the alleles show complete dominance. Okay, so here's that Punnett square. You can see here are the possible gametes from the purple cut plant, which is the dominant, right? And then it's the heterozygous, right? And then the possible from the homozygous recessive, so green potato. Okay, so you can see here. So this is going to be the purple stem and the cut leaves, purple stem potato leaves, green stem cut leaves, green stem potato leaves. So if both parents are heterozygous in this case, then you sort out all the gametes to do a more complex Punnett square. Okay, so gametes, so looking at the top, you have a parent with purple stem and cut leaves, crossed with a purple stem, cut leaf, okay, 2 heterozygous for both those traits, the dihybrid. Those gametes could be either of these, okay? So when you set them up this way, you set up all the possible gametes from each parent. So you have homozygous dominant, heterozygous, heterozygous, homozygous recessive, and then from the other parent, homozygous dominant, heterozygous, heterozygous, homozygous recessive. And you cut all the way through, and what that gives you is a very, very typical possibility that you see in these dihybrid diagrams. So when you sort out the numbers among these 16 possibilities, you find 9 that are purple with the cut leaf, 3 with the purple and the potato, 3 with the green and the cut, 1 with the green and the potato. That's a 9-to-3 to 3-to-1 ratio, and it's the typical dihybrid cross between 2 heterozygous organisms. So you've already seen interactions between alleles at the same locus, okay? Namely codominant alleles in flower color in the snapdragons, dominant and recessive alleles in tomato plant stem color, and multiple alleles in the inheritance of the ABO blood groups. So there are also cases where different loci interact to affect one phenotypic character. In the inheritance of feather color in chickens, there is an interaction between 2 gene loci. Individuals carrying the dominant allele have white feathers, even if they also carry the dominant allele for colored feathers. And birds that are homozygous recessive are also white. So let's look at that. Okay, so white leghorn chickens have the genotype capital I, capital I, capital C, capital C. Okay, the white Wyandotte chickens have the genotype lowercase I, lowercase cc. When a white leghorn is crossed with a white Wyandotte, see here, okay, all offspring are white and heterozygous. Okay, so then looking at that next generation, the offspring are interbred from the original to give another generation. The usual 9:3 to 3:1 ratio expected in this generation has been modified to a 9:3 to 1, 3 giving 13 white, 3 colored. Okay, I encourage you to pause this and really take a look. Okay, so a different interaction is shown by the inheritance of flower color in salvia. A pure breeding pink flowered variety is crossed with a pure breeding white. The offspring had purple flowers. Interbreeding these offspring to give another generation resulted in purple, pink, and white in the ratio of 9:3 to 4. Okay, encourage you to pause this and really look at that cross. Okay, we're going to talk about autosomal linkage. So this is when 2 or more gene loci are on the same chromosome. They don't assort independently in meiosis as they would if they were on different chromosomes. The genes are thought to be linked. So genes on a chromosome other than sex chromosomes are said to be autosomally linked. Linkage is the presence of 2 genes on the same chromosome, so they tend to be inherited together and do not assort independently. Okay, an example of this would be the fruit fly. It normally has a striped body and antennae with feathery arista. The gene for body color and the gene for antenna shape are close together on the same chromosome and are linked. A black body with no stripes results from a recessive allele called ebony. A recessive allele for antenna shape, called aristopedia, gives an antenna looking rather like the leg, with 2 claws on the end. So to help keep track of linked alleles in a genetic diagram, you can bracket each linkage group. So in this case, the genotype of the striped body fly with normal antennae is written capital E, capital A in brackets, and capital E, capital A in brackets, and not together, which would indicate that the genes were not on the same chromosome. So a fly homozygous for striped body and normal antennae was crossed with a fly homozygous for ebony body and aristopedia antenna. All the offspring had striped bodies and normal antenna. So male offspring were then test crossed with females homozygous for ebony body and aristopedia antenna, producing the 2 original parent types in equal numbers. The test cross gives a 1:1 ratio of the 2 original parental types, not the 1:1:1 ratio that's expected from a dihybrid cross. The dihybrid cross has behaved as a monohybrid cross. The alleles that went into the cross together remained together. The genes of any organism fall into a number of linkage groups equal to the number of pairs of homologous chromosomes. So total linkage is very rare. Almost always the linkage groups are broken by crossing over during meiosis. In the example discussed above, a male fruit fly was test crossed to show the effect of linkage because unusually there is no crossing over in male Drosophila. So pause this, take a good look at that. Okay, so during prophase 1 of meiosis, the pair of homologous chromosomes can be seen to be joined. Okay, and this is by a chiasmata. So the chromatids of a bivalent may break and reconnect to another non-sister chromatid. This results in an exchange of gene loci between a maternal and paternal chromatid. So if we look at the fruit fly cross described and test cross the female offspring. So you see the female striped body, normal antenna, male. You can see how that goes through. Okay, large numbers of parental types of flies are produced. They're in a 1:1 ratio. Okay, smaller numbers of recombinant flies are produced. These result from crossing over and recombine the characteristics of the original parents. Okay. So again, pause this, look carefully at what's happening there. So large numbers of the parental types are produced here, but the chance of a crossover taking place between the 2 loci is directly related to their distance apart on the chromosome. Okay, so now we're going to talk about the chi-square test. So this test allows us to compare our observed results with the expected results and decide whether or not there is a significant difference between them. So chi-square is the sum of the squared difference between observed and expected data, divided by the expected data in all possible categories. And this test can be used in genetics where the genotypic and phenotypic ratios have already been established for a given test population. Okay. So a gene is made up of a sequence of nucleotides, each with its own base. The different alleles of a gene contain slightly different sequences of bases. These different alleles originally arose by a process called mutation. Mutation is an unpredictable change in the genetic material of an organism. A change in the structure of a DNA molecule producing a different allele of a gene is called a gene mutation. Mutations may also cause changes in the structure or number of whole chromosomes in a cell, in which case they are known as chromosome mutations. Mutations may occur completely randomly, with no obvious cause. However, there are several environmental factors that significantly increase the chances of mutations occurring. So all types of ionizing radiation, alpha, beta, and gamma, can damage DNA molecules, altering the structure of the bases within them. Ultraviolet radiation can also do that, as well as many chemicals. So a substance that increases the chances of mutation is called a mutagen. In gene mutations, there are 3 different ways in which the sequence of bases in a gene can be altered. And these are base substitution, where one base simply takes the place of another. So, for example, CCT GAG GAG may change to CCT CTG GAG. Okay, and there's base addition, where one more extra base is added to the sequence. For example, CCT GAG GAG changes to CCA TGA GGA G. Okay. Base deletion, where one or more bases are lost from the sequence. For example, CCT GAG GAG changes to CCG AGG AG. Base additions or deletions usually have the most significant effect on structure and therefore the function of the polypeptide that the allele codes for. So those are usually the most deleterious mutations. Okay, base additions or deletions always have large effects because they alter every set of 3 bases that follows them. Base additions or deletions are said to cause frame shifts in the code. So often the effects are so large that the protein that is made is totally useless, or the addition or deletion may introduce a stop triplet partway through a gene so that a complete protein is never even made at all. So a frame shift mutation shifts the grouping of the bases and changes the code for the amino acids. A frame shift mutation occurs when nucleotides are inserted into or deleted from the DNA and they cause a shift, right? So it moves everything down. So the normal sequence is CAG, CCCACT, but if a T is added in here, that changes all the codons all the way from where it's added in all the way to the end. And that's going to change the amino acid sequence, which is going to change the protein in the end. A mutation that has no apparent effect on an organism is said to be a silent mutation. Base substitutions are often silent mutations because many amino acids have more than one triplet code. So even if one base gets changed, the same amino acid might still be coded for. Okay, again, pause these, look closely at the pictures that I have to make sure you really understand. Okay, so the gene that codes for the amino acid sequence in that B globin polypeptide is not the same in everyone. In most people, the B globin polypeptide begins with the amino acid sequence coded from the H to the B A allele. Okay, and you can see here. But in people with the sickle cell allele, the base sequence CTT is replaced by CAT, and the amino acid sequence changes. Okay. This small difference in the amino acid sequence makes little difference to the hemoglobin molecule when it's combined with oxygen. But when it's not combined, the unusual B globin polypeptides make the hemoglobin molecule less soluble. So the molecules tend to stick to each other, forming long fibers inside the red blood cells. The red blood cells are pulled out of shape into a half moon shape. When this happens, the distorted cells become useless at transporting oxygen, so they get stuck in the small capillaries, stopping any unaffected cells from getting through as well. So then a person with this unusual B globin can suffer severe anemia and can die. So sickle cell anemia is especially common in some parts of Africa and in India. Okay, albinism provides an example of the relationship between a gene, an enzyme, and a human phenotype. So in albinism, the dark pigment melanin is totally or partially missing from the eyes, skin, and hair. In humans, this results in pale blue or pink irises in the eyes and very pale skin color and hair. The pupils of the eyes appear red. The condition is often accompanied by poor vision, rapid jerky movements of the eyes, and by a tendency to avoid bright light. Mutations at several loci may be responsible for this condition, but in its classic form, the mutation is an autosomal recessive, and in individuals that are homozygous for the recessive allele show albinism. Such individuals occur about 1 in 17,000 births. However, the condition is more common in some populations, like the Hopi in Arizona, the Cuna San Blas Indians in Panama, and a different form of albinism which affects the eyes but not the skin is sex-linked. So here's a picture. A mutation in the gene for the enzyme tyrosinase results in either the absence or the presence of inactive enzyme in the cells responsible for melanin production. In these melanocytes, the first two steps of the conversion of the amino acid cannot take place. It is a transmembrane protein. It's found in the membrane of large organelles. Most of the protein, including the active site, is in the melanosome. And so tyrosinases occur in plant as well as in animal tissues. The action of the enzyme can be seen in the blackening of a slice of potato left exposed to the air. Okay, Huntington's disease is an inherited disorder that results in death of brain cells. Adult onset Huntington disease, the most common form of the disorder, usually appears in a person's 30s or 40s. Early signs and symptoms can include irritability, depression, small involuntary movements, poor coordination, and trouble learning new information or making decisions. Many people with Huntington disease develop involuntary jerking or twitching movements known as chorea. This condition is inherited in an autosomal dominant pattern, which means one copy of the altered gene in each cell is sufficient to cause the disorder. An affected person usually inherits the altered gene from one affected parent. Okay. HD is a trinucleotide repeat disorders, which are caused by the length of a repeated section of a gene exceeding the normal range. The HTT gene is located on the short arm of chromosome 4. Okay, it contains a sequence of 3 DNA bases, CAG, repeated multiple times. Okay, this is called a trinucleotide repeat or stutter. People with the adult onset form of Huntington disease typically have 40 to 50 CAG repeats in the gene. Affected individuals may have trouble walking, speaking, and swallowing. Brain cells are lost and the ventricles of the brain become larger. Okay, mutations in this gene cause Huntington disease. An increase in the size of the CAG segment leads to the production of an abnormally long version of the protein. And this elongated protein is cut into smaller toxic fragments that bind together and accumulate in neurons, disrupting the normal function of the cells. That dysfunction and eventual death of neurons in certain areas of the brain underlie the signs and symptoms of Huntington disease. Okay, here you can see the Punnett square between a mother who is a carrier and a father that is homozygous recessive. Okay, structural genes. Genes that code for proteins required by a cell are called structural genes. So structural genes give rise to enzymes and other structural components of cells. A regulatory gene is a regulator gene that's involved in controlling the expression of one or more other genes. Okay, so a repressible enzyme is an enzyme whose synthesis or rate of production is inhibited when the product that it synthesizes is present in high concentrations. So it's a way of shutting down the synthesis of an enzyme whose product is not required because so much of it is readily available. So when that enzyme product is no longer available, more of the enzyme is synthesized. An inducible enzyme is an enzyme in substantial amounts of which are formed only in the presence of its substrate or a substance resembling its substrate. An operator gene, so in genetics, an operator is a segment of DNA to which a transcription factor binds to regulate its expression. Okay. And so an operon is a cluster of genes regulated at one or more promoter site. The complete cluster of genes transcribed into a single mRNA consists of information for the synthesis of polypeptides. In an operon, genes are regulated coordinately. Either all the genes are described or none are transcribed. More specifically, an operon is a segment of DNA containing adjacent genes, including structural genes, an operator gene, and a regulatory gene. An operon is thus a functional unit of transcription and genetic regulation. Okay. The lac operon consists of a cluster of 3 structural genes and a length of DNA, including operator and promoter regions. The 3 structural genes are LAC-Z, coding for beta-galactosidase, converts lactose into glucose and galactose. Okay, LAC-Y or LAC-Y, coding for B-galactoside permease, which transports lactose into the cell. And LAC-A, coding for B-galactoside transacetylase, which we don't really know the function of. Acetylase, transacetylase. Okay. So prokaryotes like the bacterium E. coli have an efficient mechanism for metabolizing lactose. 3 proteins that are important in lactose metabolism are all encoded in a single expressible unit of DNA, and that's the lac operon. The bacterium does not waste energy expressing these proteins if lactose is not present. It only makes these proteins when lactose is available to be metabolized. A repressor, which is always present in the cell, binds to that lac operon and prevents transcription by blocking the passage of RNA. However, when lactose is present, lactose will bind to the repressor, change its shape, such that the repressor can no longer bind to the operon, and then in this case, RNA polymerase proceeds through the operon and transcribes the genes. So the lac operon consists of a cluster of those 3 structural genes. And again, that's LAC-C, LAC-Y, and LAC-A. So the repressor protein is initially produced in its active form. It therefore immediately diffuses and binds to the operator, switching the lac operon off. This prevents transcription of the genes involved in lactose metabolism. To switch the operon on, the inducer must bind to its allosteric repressor protein. The inducer is a small molecule that allosterically inhibits the repressor protein, causing it to assume an inactive form. This inactive form can no longer bind to the operator, so transcription can now occur. The inducer is named as such because it induces the system to produce the necessary enzymes. For the lac operon, allolactase is the inducer. Allolactase is a chemical isomer to lactose and is always present when lactose is. So it induces the production of enzymes necessary for lactose digestion. Okay, so here's a picture of the lac operon. Okay, allolactase is the chemical isomer to lactose. It's always present when lactose is. It induces the production of enzymes necessary for lactose digestion. Okay, pause this, take a good look at this. Okay, so in general, the number of different proteins that act as transcription factors increases with increasing size of the genome. This means that eukaryotes have many more ways of regulating gene expression than prokaryotes. So in humans, for example, about 10% of genes code for proteins which act as transcription factors. The factors can bind to a promoter region of a gene. They may increase or decrease the transcription of that gene. Whatever the mechanism, their role is to make sure that genes are expressed in the correct cell at the correct time and to the correct extent. The effects of transcription factors include the following. So a transcription factor is a molecule that controls the activity of a gene by determining whether the gene's DNA is transcribed into mRNA. A promoter is a sequence of DNA needed to turn a gene on or off. The process of transcription is initiated at the promoter. Promoters are located near the transcription start sites of genes on the same strand. General transcription factors are necessary for transcription to occur. They form part of the protein complex that binds to the promoter region of the gene concerned. Other factors activate appropriate genes in sequence, allowing the correct pattern of development of body regions. A transcription factor is responsible for the determination of sex in mammals. Transcription factors allow responses to environmental stimuli, such as switching on the correct genes to respond to high environmental temperatures. Some transcription factors, including the products of protocons and tumor suppressor genes, regulate the cell cycle growth and apoptosis. Hormones have their effect through transcription factors as well. And we know the plant hormone gibberellin controls seed germination in plants. It's a good example of how hormone can influence transcription. Okay, and it has this effect by causing the breakdown of DELA proteins. A DELA protein inhibits the binding of a transcription factor. Causing the breakdown of DELA protein allows PIF to bond to its target promoter. Transcription of the gene can then take place, resulting in an increase in amylase production. Okay, here's a model of that. Okay, that is the end. We'll see you in class.